Tissue Dynamics, a presentation by Professor Gershom Zaitchik, Faculty of Medicine, Hebrew University of Jerusalem. This current chapter is a chapter 2 of a presentation about streaming organism, and the chapter 1 was published in ResearchGate. So we are talking about some definition. Cell is the indivisible unit of the life. It's atom of life. All cells come from cells, which was formalized by Virchow. Tissue proliferative unit, TPU, is the atom of histopathology. At its origin is a stem cell. Above the stem cell are the progeny of the stem cell, which are streaming out along the unit. Stem cell division is associated with cell displacement toward the unit end, and TPU is monoclonal. So here you have the structure of such a tissue proliferative unit. It has 3 states of cells, each has stem cell, progenitor, and end cell, and these are states of differentiation of each of these cells. And the arrows indicate that as a cell changes position along the unit, it actually differentiates, enters a new state. The arrow do not push or neither pull the cells through the unit. The cells are simply streaming, and this is the essential, that in the streaming organism, cells are streamed from one place to another place, but they are not pushed mechanically. So as these cells stream, they differentiate, and once they reach the end cell, they die and they are replaced by other cells. So this determines the cell turnover of this unit. Cells are born at the stem cell, and cells are die after they reaching the end cell state. Again, once they are born, they stream along the unit and differentiate during the streaming until they become end cell. Our story starts with a zygote, which I described also in the previous presentation. The zygote is a primordial stem cell, and it then changes into a morula, blastula, and gastrula. And these are stem cell morphogenesis. And the gastrula stage is a stage where the primordial layer were formed, and we have 3 layers which are determined in the gastrula. One is ectoderm, mesoderm, and endoderm. And these layers of stem cells are the first layers where histopathology begins, because here the tissue proliferative units are created. And here you have an example. We are at the gastrula state. All these cells are stem cells, and here you have one stem cell in the endoderm starts creating a unit, and here is another stem cell at the ectoderm, which creates a more advanced unit. So these are the tissue proliferative units which determine what happens in histopathology. So here's an example. We just were here. Here was the gastrula where the stem cells start creating its progeny. And you see that next state we have the formation of one progeny, and then we have the end cell. And once the unit matures into the full all these states, the end cell starts dying, and we get a steady state where cells are formed at the stem cell stage and they are dying at the end cell stage, and the unit length remains indivisible or constant. So here's an example which ought to illustrate how you determine the state of these cells. So here in this experiment we have a unit and we irradiate this cell, and the cell dies, but it is replaced by a new cell which differentiated from the unharmed stem cell, and we get gradually a full healthy unit with a state which I call recovery. Now the next experiment, we irradiate all end cells and see what happens. So if you see such a end cell, you have to remember that at a certain moment this cell differentiated into an end cell, and this end cell then died, and therefore you see only the dying stem cell. But the dead stem cell started as an end cell and was destroyed or killed by this irradiation, and since this irradiation continues, we get a steady state solution of these units, and we have a chronic irradiation state. So the last experiment, we decide to irradiate the stem cell, and as I said, the stem cell, the progeny of this unit are progeny of a stem cell. So once you irradiate this stem cell, the entire unit dies and disappears, and it is replaced by a neighboring stem cell which was not irradiated, and we get again regeneration of an entire unit. So these were three examples which I provide as introduction to see what are the dynamic of the streaming cancer, because all cancers which we observed in our organism start as tissue proliferative units, and this was our first chapter to understand how this cancer going to behave. Enjoy.